Actin, the superstar of eukaryotic cells, takes center stage as the abundant protein, strutting its stuff, and making up to 15% of the total protein in these cells. But wait. There's much more. We have a supporting cast of actin-binding proteins, accounting for an additional 10% of the cellular protein. Now, in the muscle cells, actin and myosin steal the show, commanding over 60% of the total protein seen. They're the dynamic duo, working together to flex and contract those mighty muscles. But what makes actin so fascinating is its structure. Picture this. That actin folds into two domains, an outer domain and an inner domain, with an adenine nucleotide nestled in between. This setup stabilizes the domains. And the actin polypeptide, with its 375 residues, cleverly crisscrosses between the two domains, like a skilled acrobat. With the N and C termini located near each other. Fun fact. The two domains are folded in a similar fashion, hinting that the actin gene might have emerged from the duplication of an ancestral gene. Evolution at its finest. Well, these domains are further divided into subdomains. 2 each. Means there are total 4 subdomains of actin. Subdomain 1. Subdomain 2. Subdomain 3. And subdomain 4. Now, let's talk about actin's dance with adenosine triphosphate, or ADP. Or adenosine diphosphate, or ADP. And a divalent cation called magnesium. It's a love triangle. Magnesium acts as a bridge between ADP, or adenosine triphosphate, and actin monomers. It helps facilitate the binding of ADP to actin by coordinating with phosphate groups in ADP, and interacting with specific amino acid residues in actin. Actin binds ADP with a higher affinity than ADP. Since cells are overflowing with ADP, our unpolymerized actin is saturated with it. They're a perfect match. The bound nucleotide exchanges slowly with the nucleotide in the surroundings. But wait. There's a twist. Different actin monomer binding proteins can either inhibit or promote this nucleotide exchange. The bound nucleotide stabilizes the actin molecule, but isn't necessary for its polymerization in vitro. Oh, and by the way, ADP actin and ADP actin don't hit the dance floor at the same pace. They've got their own unique moves. By which, I mean that they polymerize at different rates. Now, let's cue the spotlight on actin isoforms. Brace yourself for a diverse cast. Most organisms have multiple actin genes, and mammals are no exception. Humans, the stars of the show, express 6 actin isoforms. Among them, non-muscle cells show off with beta and gamma isoforms, while various muscle cells boast 4 different alpha and beta isoforms. But red blood cells have their favorite. Beta-actin all the way. These isoforms bring a touch of individuality, allowing actin to interact with different proteins based on the specific needs of muscle and non-muscle cells. It's like having different dance partners for different occasions. Now, let's explore actin's role on and off the stage. In muscle cells, actin steals the spotlight, showing off its contractile function. But when it comes to non-muscle cells, actin is a multitasking superstar. It maintains the structure of microvilli. You know microvilli? Yes. These are the tiny finger-like projections on the cell surface. It's also a key player in the terminal web, which is in fact a specialized region of the cell cortex, which is located beneath microvilli and desmosomes, ensuring the cell cortex stays in tip-top shape. But that's not all. Actin knows how to shake things up, enabling macrophages to move with style through gel-sol transitions of the actin network. Imagine actin transforming from a solid gel phase to a soluble sol phase, all thanks to some cool actin-binding proteins. Well, in case you don't know, in the gel phase, actin filaments are densely polymerized, forming a stable and interconnected network of solid or semi-solid consistency. While, in the sol phase, actin filaments are depolymerized or less densely packed, resulting in a more fluid-like state. And let's not forget about fibroblasts and nerve growth cones. Their movement is facilitated by controlled polymerization and rearrangement of actin filaments. So, that was about the structure and function of actin. Now, let us move forward to the next videos to find out about the actin polymerization and actin-binding proteins.
Discover a wealth of accurate, up-to-date medical videos for in-depth learning, anytime, anywhere. Elevate your medical expertise with sqadia.com.